Welcome and thank you for listening to my talk on macromolecules, which you can read more about in Chapter 3 of your textbook. Okay, so we call them macromolecules because they're large molecules. And before I get started on that, I wanted to draw your attention to this chart, which is from Figure 3.2 in your textbook. And this chart has functional groups, and the ones that I would like for you to know are hydroxyl, which is an OH group, carboxyl, which is a carbon double bonded to an oxygen and single bonded to a hydroxyl group, amino, which is a nitrogen bonded to two hydrogen atoms, and phosphate groups, which are at the bottom. I'm going to try to show you if I can drag the screen down. So this is a phosphate group. It's a phosphorus atom, single bonded to 3 oxygen atoms and double bonded to 1 oxygen atom. So I'd like for you to know those. And you can skip the second one, carbonyl, for now. Okay, so we're going to go through the basics of macromolecules, stuff that's general and that applies to all of the different classes of macromolecules. And then I'm going to take you through each of the 4 classes of macromolecules separately. So the 4 types are proteins, nucleic acids, carbohydrates, and lipids. The first 3 on the list have a lot in common, and then lipids are kind of the oddball, but they still have some things in common with the other groups on the list. So macromolecules, as I said before, are really large molecules that are put together from many smaller components called monomers. And when you put a bunch of monomers together into a chain, you end up with a polymer. So each of the different classes of macromolecules has its own monomer and polymer, and the monomers and polymers have names that are unique to each class of macromolecule. So that is going to be one of the takeaways from today, is the name of the monomer, the name of the polymer, and the name of the bond that forms between the monomers within each class of molecule. And then, but first I'm going to go through the things that are similar across all of these types of molecules. So all macromolecules are assembled in this way, including lipids. So when you add a monomer to another monomer of any of the types of macromolecules, you lose a hydrogen atom from one of the monomers or subunits, and you lose a hydroxyl group from the other monomer that's being added, which is in effect losing a molecule of water. So this type of reaction that builds macromolecules is called dehydration synthesis, and you will want to know that. And for every monomer that is added onto the chain, or for every bond that is formed, a covalent bond that is formed between 2 monomers, you have a separate dehydration synthesis reaction, and you lose 1 molecule of water. So in order to form this bond and this bond, 2 dehydration synthesis reactions had to happen, and for each one, a molecule of water was lost. Okay, so dehydration synthesis puts macromolecules together, and to break down macromolecules, the process is known as hydrolysis. And hydro refers to water, lysis means to break, and so hydrolysis is just adding a molecule of water to break off or to break the bond between 2 monomers of any polymer of macromolecules. So for each bond that's broken, a separate hydrolysis reaction happens, and it takes 1 molecule of water. And so if you look down here, it's just showing a hydrogen being added to 1 monomer and a hydroxyl group being added to the other. So this is basically the reverse of dehydration synthesis. Okay, so we're going to just launch into proteins. Proteins are complex. The monomers of proteins are called amino acids. A polymer or a chain of amino acids is called a polypeptide. So that's the polymer for proteins. This is just showing a chain of different amino acids. And I'm going to show you the parts of an amino acid. And this is going to be another takeaway for each of these classes of molecules, is to know the parts of each type of monomer. So an amino acid consists of a central carbon atom, which is attached to a hydrogen atom, which should be added to this list, an amino group, which is NH2, a carboxyl group, which is COOH, and then a functional group or a side chain, which we call R. And the side chain or the R group is different for every amino acid. There are 20 different amino acids, and each one has a different R group or side chain, and that is how you tell the difference between one amino acid and the next, is the R group. Everything else is the same except for that R group is unique to each amino acid. So this is a picture of what I just told you. There's a central carbon atom attached to a hydrogen atom. It's attached to an amino group and a carboxyl group, and then this is showing the R group, the wild card. Okay, so the other thing I wanted to point out about proteins is that the covalent bond that links two amino acids together is called a peptide bond. A peptide bond, again, is formed through a dehydration synthesis reaction. Okay, so proteins are structural. So another thing that you'll want to know about each group of molecules is the function. Proteins can be structural. Proteins can also be enzymes that facilitate or catalyze biological reactions, make them happen at a speed that's fast enough for the organism to maintain homeostasis, which we've talked about this semester already. So the order of the amino acids in the polypeptide chain determines what protein you're going to have. It determines how the protein is going to fold together and eventually have a three-dimensional shape, and the three-dimensional shape of a protein determines the function and is necessary for that protein to carry out its intended function. So I'm going to try to go through this as quickly as possible. There are 4 levels of structure in proteins. I'm going to want you to know this. I'm going to hit them very briefly here, but you can read more about each of them in your book. So you have the primary structure, which is the order of amino acids in the polypeptide chain. The secondary structure is the initial folding of the polypeptide chain. You have 2 types. You have beta pleated sheets, and then you have alpha helixes. You can look at this more in your book. The tertiary structure is the final 3D or three-dimensional shape of a protein molecule after all the folding is finished. And the types of interactions that cause the secondary structure and the tertiary structure to happen or be possible are weak interactions such as hydrogen bonds, which we've already talked about once this semester, and disulfide bridges. And so this three-dimensional structure of the protein that's necessary for the protein to carry out its function is held together by these weak interactions, and I'll tell you a little bit more about how that affects proteins in a minute. And so finally, you have quaternary structure, which not all proteins have. The quaternary structure is if there are 2 or more polypeptide chains in a protein, and the quaternary structure is just how the multiple polypeptide chains fit together to form the protein. But if you only have one polypeptide chain, then your levels of structure stop at number 3, tertiary structure. Okay, so I was talking about how the three-dimensional shape of the protein is held together by weak interactions. Well, the way that this affects proteins is that changes to the environment of a protein, such as high temperature or low or high pH, can affect the way that the protein's shape is held together and cause it to unfold, which is known as denaturing. And if a protein becomes denatured, it loses its three-dimensional shape and loses its function. It becomes inactive. You can watch this video at home if you wish. Okay, so this is just a picture from your book from section 3.2 showing a protein. And proteins, if they're enzymes, have active sites that bind the reactants or the substrate molecules to catalyze or facilitate a reaction. So if this three-dimensional shape is lost, the shape of the active site is lost, and so the active site doesn't fit the substrate any longer. Okay, so I'm going to move on to nucleic acids. Okay, so nucleic acids are, the function of a nucleic acid is that they have the genetic code for an organism. It's the DNA and the RNA. Nucleic acids are really long polymers that store this genetic information. The monomers of nucleic acids are called nucleotides. Okay, so each nucleotide has 3 parts. So it has a 5-carbon sugar, a phosphate group, and then a nitrogen-containing base, often referred to as a nitrogenous base. And there are 5 different types of nucleotides, and we'll talk about that really briefly. But just like the order of amino acids in a chain determines what protein you have, the order of nucleotides in a polymer of or in a polynucleotide determines what proteins are built and what the organism is going to look like and how it's going to function. Okay, so this is just a picture, I'm going to move this up, showing the structure of a nucleotide. And so this is the sugar, this is the phosphate group, and then this is the nitrogen containing base. So in RNA, this sugar is ribose. In DNA, the sugar is deoxyribose, and that's one of the differences between the two. So that is what your nucleotide structure looks like. Down here are the different nitrogenous bases. I'm not going to ask you to remember these structures because they're pretty complicated, but I do want to point out, you notice that 2 of them, adenine and guanine, have 2 rings in their nitrogenous base. This part right here is this right here. So the nucleotides that have 2 ring structures in their nitrogenous bases are purines, and the purines are adenine and guanine. And then these 3 down here, cytosine, thymine, and uracil, each have only 1 ring in their nitrogenous base, and those are called pyrimidines. You should read about this in your book. And then I would like for you to know which ones are in DNA only, which ones are in RNA only, and which ones pair. And I would like for you to read about that in your book, okay? So let's see. Okay, so we talked about the 2 types of nucleic acids, deoxyribonucleic acid and ribonucleic acid. RNA and DNA, another difference is that DNA is a double-stranded helix, and so you have the base pairing, which I'd like for you to read about in your book. These are hydrogen bonds represented here, the dotted lines between the pairs of bases. And then RNA is a single strand. RNA has uracil only, and DNA has thymine And